Welcome you all for the next lecture on inorganic chemistry of life, principles and perspectives. Let us go through a recapitulation of what I have taught in the previous class. In the previous class, the main theme of whatever I talked about is the metal ion is present in the enzymes as a coordination complex which is surrounded by the protein, and as a result of that the metal ion properties do change with respect to the type of protein that you have, protein conformation, protein sequence, all different kinds of details of the protein as compared to the simple coordination complex or an ion in an aqueous medium. So, either of these if you compare, and this is a boon in disguise, because by changing the protein surroundings and the nature of the protein itself, you can change altogether properties of the enzyme. Okay, having been talking a lot about the protein side chains etc., I thought it is appropriate for those who may or may not know more details about this protein. So, I thought I will give some basic details of these very basic details of these proteins. And okay, so let us look at first of all starting from the amino acid. So, you see the amino acid is also referred as alpha amino acids. You have a carbon center with one hydrogen and there are 3 substitutions, of which one is the carboxylic group, other is the amine group, and this is common for all amino acids. And what differs from one amino acid to the other is the presence of this particular group. Okay. So, this particular group is R group. So, this is just referred as R, in other words the side chain. So, this side chain varies from one amino acid to the other among the 20 naturally occurring amino acids. Of course, today 21 is also added, which is selenocysteine in the literature. Otherwise, we can take just as 20 amino acids. So, that is not a problem. And so, you can see that the same thing as shown over there, and the carboxylic group losing a proton, amine group gaining a proton, this is referred, such kind of a structure is called the Zwitter ionic structure, and this structure of this Zwitter ionic or otherwise is dependent on the medium pH. So, the pH of the medium will have a control whether you have a COOH or COO minus, NH2 or NH3 plus. So, generally these are represented by the kind of a structure, this Zwitter ionic. Now, the different amino acids come out because of the change in the R group. So, let us look at the next slide. So, in this slide, so what we are seeing, having known that there are so many different kinds of amino acids are there, and these amino acids together can form, let's come once for these set of the amino acids that we have. We have glycine, we have alanine, we have valine, leucine, isoleucine, methionine, proline, phenylalanine, tyrosine, tryptophan, and aspartic acid, glutamic acid, asparagine, glutamine, cysteine, serine, threonine, histidine, lysine, arginine. I have already explained you the kind of a side chains that are present in 2 lectures prior to this. If you look at those slides, you will find what are the side chains that you have. I have given in the form of side chains the non-polar, polar, polar with charged, positively charged, negatively charged, all this. I have also explained those 6, 7, 8 different amino acids which are capable of binding to the metal centers. This is already talked to you. And in the protein literature, it is commonly the amino acids are referred by a 3-letter code or a 1-letter code, and it is better that you are familiar with these ones. The glycine is written as GLY, alanine is written ALA, valine is written VAL, leucine is written LU, LEU, etc., etc. Aspartic acid is written as ASP, etc. Now, if you look at this 1 code letters, then you would see that the glycine is given as G, alanine C is A, valine is V, leucine is L, etc. Now, if you come to glutamic acid, it cannot have G because G is already there. So, they have given E as the one. Aspartic acid A, that is fine. Aspargine A is already given. So, therefore, N is given here. So, glutamine and cysteine is C, which is not used earlier. So, serine S, threonine T, histidine H. So, therefore, you have a 3-letter code and 1-letter code, and when you write a protein sequence, you start using the one single letter code, and when you write small peptides, you generally use the 3-letter code, and that is all the difference is. Now, let us go back to the slide. As I said, these are the amino acid side chains, and the side chains differ as polar, non-polar, charged, positive, negatively charged, and they are all having ligating centers. They can bind to the metal centers. Now, having known individual amino acid, let us look at the peptide. So, peptide or protein. Protein is what? Protein is the one which is formed with the peptide bonds. So, protein is the one which is formed from the peptide bonds. How is a peptide bond is formed? Peptide bond is formed between the two amino acids. So, carboxylic end of the one amino acid reacting with the amino end of the another amino acid, and there is an elimination of water, which is called dehydration reaction, and as a result of that you will get a bond CONH. This bond is referred as the peptide bond or amide bond. If it is in a protein, it is called peptide bond. If it is in simple amides, then they call it as an amide bond. So, they are one and the same. So, amino acid 1, amino acid 2. So, you can see R1 and R2. Sometimes this R1 and R2 can be the same, or R1 and R2 can be different. And as you can see that the carboxylic of this and the amine of this interacting together through the loss of water. So, you have an amine left over here and a carboxylic left over here. So, therefore, this is referred as the amine terminal or amino terminal, carboxylic terminal or carboxylic C terminal. So, N terminal and C terminal. Let us look at a little bit more or longer, a bigger case. So, we have 4 different amino acids. The amino acids serine, valine, tyrosine and cysteine. So, these ones, if you see here, the carboxylic of this amino acid reacting with the amine of this amino acid, carboxylic of this one, amine of that, carboxylic of this, amine of that. So, if you have a condensation here, condensation here, condensation here, loss of water, loss of water, loss of water will give what? 3 peptide bonds, one here, one here, one here. So, this whole thing has got 4 amino acids, which is also referred as a tetrapeptide. Amino acid 1, amino acid 2, amino acid 3, amino acid 4. How do you recognize the amino acid? From the C alpha center. So, C alpha center you can easily identify these ones. Okay, this is a tetrapeptide. So, if you extend, it becomes a polypeptide. So, from several. So, now what you say, each one of these amino acid is called a building block in these. So, you have a building block. Now, after this is formed, what you have? This end you have amine, this end you have a carboxylic, but otherwise there is no other amine and carboxylic. You only have CONH. So, therefore, what is important? Only the side chains. In a huge protein, let us say having 100 amino acids, 200 amino acids, 1000 amino acids, then what is important? The important thing is that side chains in all those cases. So, let us re-look at this is a tetrapeptide, it has a peptide bonds over here and side chains. In a huge polypeptide like protein having 100, 200, 1000 amino acids, these will become terminals and the entire protein will have the side chains coming up. So, the same thing as I talked to you earlier, you start with the amino acid, combine with one more amino acid, combine with one more amino acid, combine with one more. So, you can start stitching the amino acids together to form a peptide bond, and the peptide bond connects these amino acids. So, here you can see one such polypeptide having 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 amino acid residues. So, you can call this as a decapeptide. So, in this you have a glycine, leucine, etc., all are there. So, in the junction to these is a peptide bond, in junction to these peptide bond. So, you have left with one amine part and one carboxylic part. So, the amine part is referred as the N terminal, carboxylic part is referred as the C terminal. This is important because later stage we will come across the N terminal and C terminal situations where we talk about what is happening at this over the other. Now, you have taken the amino acids, you connected them together like you take flowers and make a garland. So, let us take different kinds of flowers with a different nature or flowers with a different kind of a colors. Then you take flower 1 with red color, then you put next one is a green color, the next one is maybe yellow color, or again go back to red, etc. So, you are connecting them together like this one. So, this polypeptide is nothing like a nothing but like a garland which is made from flowers of different colors or flowers of different types. Now, such a polypeptide it converts into some kind of a matured level of the protein, which I will come in a while. This is called structure. So, the structure of this the protein is formed, and this is really ready for any function. So, I will explain the things connecting these ones. So, an amino acid, a polypeptide to a protein, and that is how we move from one to the other to the other. So, this is just simple garland, but this is not that, this is a decorated one. So, these decorations are coming from where I will explain in a while in this. So, in order to explain the protein structure that you have seen on the previous slide, or in other words I referred it as a decoration, and we need to introduce the 4 parameters. One is the primary structure, the other is secondary structure, third is tertiary structure, fourth is quaternary structure. So, these are the different stages of this protein, and we will go through this. So, the primary structure is the one where you connect the amino acids together to make a polypeptide. The secondary structure is the one where such a polypeptide will not stay like a long thread, there will be some kind of a rearrangements, structural rearrangements, and these structural rearrangements in various forms I will explain in a while. And such structural rearrangements taking shape in the entire protein, and the protein takes a topological structure is called the tertiary structure, and such tertiary structured units join together in a particular symmetry or symmetry element will result in a quaternary structure. I will again explain these things as we keep going through the next few slides. Okay, let us look at the first part of the structure, which is called primary structure, the protein. So, what the primary structure the protein has got? This is coming from one amino acid, second amino acid, third amino acid, etc., etc. So, what do you have? You have a C alpha center, another C alpha center, 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 and each one of these have got a peptide bond. You see that CONH, 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 CONH. So, this whole thing is referred is the protein chain. So, A is connected to B, is connected to C, is connected to D, etc. So, that is that kind of a sequence is referred as the primary structure. So, in a protein, when we know primary structure means we know the order in which the amino acids are connected. There are ways and means by which one can do that. This is not the stage where I will explain. I would explain some other stage, because you take a protein, you hydrolyze, you start detecting, you can find selectively hydrolyze and find which is the first amino acid, which is the next amino acid, which is the third amino acid, etc., etc. These are all very well known in the literature, but those are outside the scope of this particular course. Let us not too much worry about it. So, here is a particular structure of the protein taken, primary structure of a bovine serum albumin. So, it is a serum albumin protein from the bovine source. So, bovine is cow, the cow source, and serum is blood, and the albumin is the albumin like protein, and in that you have a chain structure shown here. you have each amino acid is connected here, and this number shows the 5th, 10th, 12th, 15th, 20th, etc. and there is another one. So, this is just to indicate the primary structure, enough, this much information. This is a primary structure, and now once the primary structure is formed, you can have the secondary structure of the protein. So, what is the secondary structure? So, this will bring stable arrangement of the amino acid residues in the backbone to give some kind of a structural pattern, okay, and that will bring to the local conformation too. So, the secondary structures which are commonly seen in proteins are two major ones. One is the alpha helix, other is the beta sheet. The third one is left out portion, which is called the random coil kind of a portion. So, means no specific structure, it is open kind of thing. So, alpha helix, beta sheet, and then the portions which are connecting these ones. Now, we will look at each of these, what is alpha helix, what is beta sheet, etc. In the next few slides. Okay, alpha helix, as you can see, is that in this particular protein we have seen earlier a polypeptide. Now, twisted the polypeptide like this and goes this way and goes this way and goes this way. Why should it twist? So, this twisting, as you can see, there is some connectivity here, and there is some connectivity here, and there is some connectivity here. So, that means these connectivities are dominating. So, these connectivities are dictating this particular polypeptide chain to take this kind of a shape, and this shape is referred as the alpha helix. So, what are these connectivities? These connectivities are CO is carbonyl, NH is the NH moiety. So, each peptide amide bond has got CONH. So, CO of this with NH of this, CO of this with NH of this. Okay. So, similarly of this. So, you have CONH kind of a hydrogen bonds. Where are these hydrogen bonds? If you take nth residue and to the n plus 4th residue, these two are connected, okay, and such a kind of things are found in the alpha helix. And the one which is shown here is a right handed alpha helix, because we are talking about all the L amino acids. If you go to D amino acids, the situation will change, let us not too much worry. And what is the, you see, when you go from here to here, you are bringing a kind of a bend. So, this bend is also referred as a torsion. So, on some bonds you have to twist and torsions. So, if you do such kind of a twist or torsion, then you will get this bend, and those angles are given over here. So, one of the angle is CC alpha, other angle is NC alpha. Okay. So, these are the two angles that you get, because these two bonds are the single bonds. So, the single bonds will have a rotation. Because of these rotations, this kind of a structure takes. So, in this alpha helix, there is 3.6 residues. Each helix turn has got 3.6 residues. Okay. And there are 14 atom hydrogen bonds in this. If you start counting between the CO and the NH of that, you will find. So, the hydrogen bonds are shown over there with more clarity here. So, this is very commonly found in many proteins, a large number of proteins. Okay. The example that is shown here on the right side is bovine serum albumin, you can see this. So, the same thing, if I put a color, it will look like this. There are different kinds of colors because they are coming from different regions. You do not need to worry at this stage why the color is different. So, all that you need to see is you can see the spiral kind of thing. So, this spiral is nothing but helical. So, these helicals are alpha helical. So, as you would see, the total protein is dominant by the helical structures. The small threads, you can see, these small threads have no specific structure. They can be called as a random coil kind of structure. And this structure anybody can see in the PDB protein data bank with this number. If you go, you will get this particular protein structure. You can get this one too. Okay. So, I hope you understand the one of the secondary structures is alpha helix, and the alpha helix is stabilized by the CONH hydrogen bonds of n to n plus 4th, and that is the driving force. And this hydrogen bond, as you know, it stabilizes the structure, because the hydrogen bond when it is formed, there is a the energy being released. Okay. So, delta H is minus. So, therefore, total delta G will come to the minus favorable. So, therefore, such structure is formed favorably in the proteins. And go to the second category of this mentioned, beta sheets. Beta sheets are formed not by a twist, they are just in their stagger, in their zigzag kind of a fashion, but between one such unit another such strand, you can say. So, therefore, you have a CONH, another CONH, another CONH, CONH this side, you have all these kind of things. So, such kind of structures are called the beta sheets. So, the beta sheets, you can see the N for this thing, this side is N, this side is carboxylic, and for this strand, this side is nitrogen or N terminal, this side is C terminal. So, therefore, N terminal to C terminal and C terminal to N terminal. So, that kind of a thing gives you the sheet type of a structure, which is called the anti-parallel beta sheet. On the other hand, both the N to C and N to C, or C terminal, N terminal, C terminal, N terminal, they are exactly aligned. It is called parallel. So, it is called parallel beta sheet. Just because of this parallel and anti-parallel, obviously you will find the corresponding sheet has a different properties. As you can see that this looks quite different from this one. This is coming from the parallel one, this comes from the anti-parallel one. So, parallel beta sheet, anti-parallel beta sheet kind of a structures. So, what I have been telling you about the beta sheet structures, parallel beta sheet and parallel beta sheet, anti-parallel beta sheet, and this only the alignment is N terminal to C terminal and C terminal to N terminal here. On this side both are aligned, both the N terminals and both the C terminals are aligned together, and this also stabilizes the structure, and this is also found in number of proteins too. You can see in a little better way here this zigzag repetitive structure, and you have seen the hydrogen bonds between the neighboring polypeptide chains. So, this is referred as the structure which resembles the pleats, okay, the pleat kind of a structure. Okay. So, therefore, two types, anti-parallel and parallel. There is one example is shown over there. Okay. This protein has hardly any kind of a helix, and it is mainly has sheets, and these sheets are shown over there here. The sheets in the form of green color, in the blue color, light blue color, and the orange, red, etc. You see that. So, but all of them are beta sheet kind of structures. So, this is a protein which is lentil, which is from lectin protein, is a concavalin A. Right now, you do not need to worry about the name of this protein. It is a beta sheet structure is dominating, and this can also be obtained from the protein data bank. Protein data bank PDB number is the number is given here, protein data bank. So, if you go into the internet and run this number under the protein data bank, you will get this structure. You can see that. So, this structure is primarily the beta sheet structure. Okay. Is it that you should have in a protein all alpha helices and you should have only beta sheets? That is not correct. There are proteins where both the alpha helical or beta sheets both are present too. You can see an example here. So, this protein has got not only alpha helical structures, but also beta sheets. This is primarily alpha helical, this has primarily beta sheet, but this one has got both of these too. Now, so having seen these alpha helical beta sheets, that means they are folding the protein in various forms. So, once you fold the entire protein, you get into a kind of a topological structure, and which is in turn stabilized by secondary structures, and the secondary structures are dictated by the protein sequence that we have and the turns that it can take place. So, therefore, in a tertiary structure you have a topological features of the total structure. So, this is shown as a tertiary structure. So, tertiary structure is a superstructure having its subset is the secondary structures together, and the primary structure of course is involved. Okay. So, several secondary structures together gives the tertiary structure for the total protein. The example shown over here is a myoglobin protein. Of course, we are going to look into the details of this at a later stage, and this is the portion where you have a heme containing portion. So, this is not the appropriate time to highlight the heme part of it, but otherwise you can see this is the tertiary structure of that. Okay, let us look at the quaternary structure. Now, you have looked at the tertiary structure in the previous slide for the myoglobin, and where it has one polypeptide chain and it has interior heme protein. Now, you take 4 such units and try to arrange. One of this is this light orangish and a bit darker brownish orangish kind of color, and then you have a pink and darker pink. So, there are 4. So, these 4, each one of these is the polypeptide chain that I have shown you in case of myoglobin 10. So, therefore, you have put the 4 of these, but these are put in a particular specific manner. So, you have a symmetry related. 2 of them are towards us or towards us, and 2 of them are towards the back side. So, therefore, you have a structure which is being built in the form of a symmetry. So, this is symmetrically based arrangement of tertiary structures is nothing but a quaternary structure. In some proteins, the tertiary structure and quaternary structures are same subunit, only one subunit. In some proteins, there can be 2 subunits, there can be 3 subunits, there can be 4, there can be 5, there can be 6, variety of things are there. So, therefore, when you have more than one subunit, they are arranged together in the form of in a particular symmetry and results in a quaternary structure too. Another example is shown over here. You can see there is a 1, the 2nd, the 3rd, the 4th, the 5th and 6th, and there are 6 more on the back side. So, this protein has got 12 subunits, and the 12 subunits of which you can see the top 6 and the bottom 6 on the back, and these are also coming from the metalloprotein called glutamine synthetase, where manganese and magnesium ions are present, manganese and magnesium ions are present. So, this is again the quaternary structure of another protein. Now, what have we learned? The primary structure, the secondary structure, the tertiary structure and the quaternary structure. So, all of these obviously are important. Okay. Now, let me put them together in one slide and explain you. So, what do you see on the left end, left extreme is shown like A1, A2, A3, A4 means amino acid 1, amino acid 2, amino acid 3, they are connected, that means it is a polypeptide. So, it is a polypeptide, they are connected in this particular fashion, therefore, it is a primary structure. So, primary structure or sequence, one and the same. So, the amino acid residues are connected in this fashion. The secondary structure is the one where in this particular example we have shown alpha helical, but you can have beta sheet also. So, which I explained you. So, they take these turns because of stabilizing forces, and to make this you have to make a twist, and which is some dihedral angle twist, and these twists are made with respect to NC alpha, CC alpha. Okay. So, CC alpha and NC alpha, these two are single bonds, therefore, single bond rotation does not cost much energy. Therefore, that is what that energy is supplied by the stabilization of the hydrogen bond. So, hydrogen bond gains the energy and twisting requires the energy. So, there is some expenditure, there is some gain. So, therefore, net there is a gain in these ones. So, therefore, secondary structures are built, and tertiary structure is this kind of a secondary structures running across the entire protein is called the tertiary structure, and such proteins are joined together in a particular kind of a symmetry, and that results in the quaternary structure. So, secondary, primary, secondary, tertiary, quaternary is, as an example, only one is shown secondary structure, but you can have other ones also. This is protein structure hierarchy. This is very important because we will be later on talking about the protein function, everything. The protein function comes not from primary structure, not from secondary structure, it comes from the tertiary structure and can even come from the quaternary structure, but not from primary and secondary, the protein functions coming into these things. So, in this lecture primarily I have focused on explaining you what is an amino acid, what is a peptide bond, what is a primary sequence, what is a secondary structure, what is a tertiary structure, what is a quaternary structure. And I have also told you, just by having the primary structure and secondary structure, function is not come, function comes from the tertiary structure and quaternary. In some case polypeptides or proteins, tertiary structure and quaternary structures are same, where only single polypeptide chain is there, otherwise they are different. So, you can have tertiary structure showing the function, quaternary structure showing the function. So, in the next class we will look at the other biomolecules like nucleic acids, etc., and we will also look at the processes like protein synthesis and other biological parameters. Thank you.